This video is key concept for chapter 4, Bio 181 on eukaryotic cells. So eukaryotic cells. Eukaryotic cells in the domain eukarya, all of these cells have a nucleus and extensive internal compartmentalization. So they're really much larger than prokaryotic cells. So outside, what I'm drawing here when I draw a cell, this is always the cell membrane. So again, every cell has a cell membrane. That's what I'm drawing on the outside. And then here, every eukaryotic cell also has a nucleus. So a nucleus is also a membrane inside, it's a sphere, and inside the nucleus is where you're going to find your DNA. So the DNA is protected in a eukaryotic cell in the nucleus. So also you're going to find lots of other compartments inside of the cell, different shapes and sizes that are going to have specialized functions. So we're going to go through those. So the nucleus, again, the nucleus in the cell, okay, it's often the largest membrane-bound compartment in the cell. So here's our nucleus. The nucleus is actually going to have some holes in the surface. So this is a sphere within a sphere. Okay, so this is the outer cell, right? This is, again, the cell membrane on the outside. And then inside is a second sphere, so a smaller ball. And there are little holes in the surface. And stuff can go in and out of these holes. So it can go in and it can come out. But again, the main idea of the nucleus is that inside we have our DNA. Okay, our DNA. Now the nucleus is actually a double membrane phospholipid bilayer. So for what it's worth, there's that. Okay. It's 2 layers thick and it's made up of the same stuff that the cell membrane is made out of. And we'll talk more about that in the next chapter. So the endomembrane system inside of the cell. So again, I'm drawing the cell again. Here's my large cell outside, cell membrane. And I don't mind that I keep telling you that over and over again because it's really important that you know that. Every cell has a cell membrane. Then inside, I'm going to draw the nucleus again. So here's our nucleus. And then there's a way that the nucleus is connected, physically connected to a series of other things. So right inside the nucleus, there's actually another enclosed compartment called the endoplasmic reticulum. Some of that is actually got these studded ribosomes on it, which is why it's going to be called rough. So this is the rough endoplasmic reticulum. And I'm just going to abbreviate ER. You should write it out, but just because of the pen and time, I'm just going to point there. That's continuous with the nucleus. So the membrane actually is connected to the nucleus. Okay? So things can travel from the nucleus in here. Then there's also part of this endomembrane system, which I'm going to draw over here, which is also the endoplasmic reticulum, but it's smooth. It does not have any bumps on it. So this is called the smooth endoplasmic reticulum, ER. And they have different functions. So the rough ER, the function of the rough ER is to synthesize proteins. And that just means it makes proteins. Okay? It makes proteins because it's covered in these dots, which are ribosomes. And ribosomes are little complexes that make proteins. Okay? So these little things here are ribosomes. Now the smooth ER, I always think of it as smooth making like fats or lipids, right? So smooth, fat, butter, it's smooth. So it's going to make or synthesize. Synthesize is just to make fats or lipids. Lipid is a big category that includes fats, and it's also going to make complex carbohydrates, which are the bigger ones. So complex carbohydrates, Okay. So just to point out here, as a reminder, we had 3 types of macromolecules that were made out of, subunits, repeating monomers. Proteins was one of them. Lipids, that was another category. Okay, this one didn't really have true monomers, but it was a biological macromolecule. And carbohydrates, okay? So this is where those are made. The proteins are made in the rough ER, the lipids are made in the smooth ER, and the complex carbohydrates are also made in the smooth ER. Where are the nucleic acids made? Well, let me ask you, what's in the nucleus? DNA, right? So this is where DNA is made. DNA made, okay? So now we know where the lipids are made, where the carbohydrates are made, the complex ones, where the proteins are made, and where the DNA is made. Okay, so just to finish this up, there's a couple more things, a Golgi and lysosomes. So Golgi apparatus, this is often shown in kind of like a pink color. That's why can't I find pink? Oh, I don't have a, Right, now is not the time. So I'll just do it over here. This is the Golgi. Kind of looks like a stack of pancakes on the side. So it's the capital Golgi. The G is capital because it's someone's name, the name of the scientist that found it. So Golgi, Italian. And this packages, so it packages proteins, okay, and other things like carbs and lipids. So it can package them, it can also modify them. So I'm going to say actually packages and modifies. Now, it's part of the endomembrane system, which means that say a protein is made, and I'm going to go back to this rough ER over here. Part of this could just kind of come off as a vesicle. It can move through the cell. It can merge with the Golgi complex. And then the contents that were in it, so say there was a protein that was made over here, it's brought over to the Golgi, and then it's put in the Golgi, then it moves through the Golgi. As it moves through, it's modified. It actually might even be modified by adding something like a lipid to it. So you might take it and add a lipid to it, or you might even add a carb to it as well. So it's going to be modified. It's going to move through. So now here's my modified molecule. And then that molecule, once it's done, can be either exported out of the cell, so it might dump the contents outside and go into the bloodstream, say if it's a hormone, or it might take the contents and it might take it back over to another organelle. So this is a connected system. This transporting object here is a little sphere, is called a vesicle, and it moves things from one place to another. It might also say move something from the smooth ER over to the Golgi. The last thing in this endomembrane system is called a lysosome. So let's see. Why can't I get different colors for crying out loud? Okay, so lysosome. Just do blue again. So here's my lysosome. Lysosome. And it has digestive enzymes. So it contains digestive chemicals or enzymes that break things down. All right, so maybe you get a big pile of food, something's brought into the cell. To digest it, it might be in a vesicle of its own that then would merge with this vesicle. Then the digestive enzymes break that down. So that's just one more component, right? So it can merge with other things. So all of this together is the endomembrane system. Okay. This is from the book, much nicer than my picture, right? But basically, just to orient you, up here, this is the nucleus. Notice the pores. This is how things get in and out. Right next to the nucleus, here's this rough endoplasmic reticulum. It's rough because it has these ribosomes on it. You can see these little studs, okay? And each of those make proteins. So it's going to actually make a protein. The protein's going to go inside here in the internal space, and then a vesicle will form enclosing the protein. It's going to transport it. So here's my transport vesicle. It's going to go to the Golgi apparatus. Now it's going to dump the protein in here. It's going to move through the Golgi. It's modified. A new vesicle forms. That vesicle then goes somewhere. In this example from the book, it happens to be a secretory vesicle. So it's going to dump it outside into the extra or outside cellular fluid. And then over here again, this is the smooth ER. It's kind of a noodly thing in 3 dimensions. Similarly, a vesicle could come off of this and then go through the whole scenario. Okay, a couple other things. These are not part of the endomembrane system. This organelle is very important though, the mitochondria. So I'm drawing the organelle. This is inside the cell. So this is, gosh, okay. I'm gonna go over here. Here's my cell on the left. This is my cell, okay? And then here is my mitochondria, and that's what this is, just so you know. Okay, so this is the mitochondria. Okay. So it's much smaller, and I've just blown it up here. So this is what I'm looking at. The mitochondria has an outer membrane. This is the outer membrane, outer. And then it also has a folded internal membrane. So it's 2 membranes thick. It's double membraned. Okay. So this is called the internal, this is the inner membrane, inner membrane. Okay. We're going to go into this in much more detail in a future chapter. What you need to know now is that the purpose of the mitochondria is to make ATP. Now, we're going to also learn about that in a future chapter, but I just want you to know that word now. So ATP, this is the energy of the cell. So you break down glucose, which is a big sugar molecule relatively, into these really small pieces. And then ATP is what's actually used to power all of the little things within the cell. So it's like, you know, glucose is a gold brick. You're not going to take a gold brick and go buy a soda pop with a gold brick. You have to break it down into smaller energy. That's what the mitochondria does. And the mitochondria is present in both, I'm just going to emphasize this here, this is a common misconception, both animals and plants, okay? And plants. All right. Chloroplasts. So chloroplasts are only going to be found in plants, okay? Or protists, which we don't really cover that much in this class. So again, this is my organelle. Here's my plant cell over here. Inside it, you're going to find an organelle. That's what this is. Okay, this is the chloroplast. Okay, that's this thing. The chloroplast also has internal compartments. Kind of look like a stack of pancakes like this, okay? So when we draw it, or when I draw it, I'll often draw it like this because it's just going to represent more accurately the true shape of this organelle. What's the function? So the function of the chloroplast is photosynthesis. So photosynthesis. And photosynthesis is to take light energy, okay, and to take this light energy and to store it in sugar. So it basically makes sugar or glucose. Okay, glucose. And I guess I should back up. So I'm using this word here, photosynthesis. That's important for the chloroplast. For the mitochondria, this process to make ATP is called cellular respiration. It's called cellular respiration. Okay. Right. So you should also associate that with the mitochondria. Okay, I think. Oh, the last thing is, so how did the mitochondria and the chloroplast arise? How did they evolve? Through a process called endosymbiosis. So endo is inside, sim is the same, bio is life. So basically what this is saying is that at some point a long time ago, we had a free living cell, right? And then we had another free living cell here, okay, which is then going to get engulfed and it's going to go inside of the big cell. Let me draw the big cell over here. So the big cell, oh, this should be in blue. Okay. So over here in blue, okay. And instead of digesting it, so the blue cell is probably trying to eat the red cell and get energy out of it, but it happened to just stay alive inside. And so now inside of the blue cell, we have the mitochondria. And similarly, we expect that the same thing happened where there was another free living like cyanobacteria, which is a bacteria that's capable of photosynthesis. It was also probably consumed a long time ago in the past. And then we ended up with a cell with chloroplasts inside, sunny green, and mitochondria. So that would be the modern plant, okay? And this process of bringing in this external organism and then living together and functioning together is called endosymbiosis. The picture of that I have here is a little bit nicer, right? This is the original bacteria that was free living. A long time ago, it got engulfed and it became the mitochondria. The cyanobacteria over here became, after being engulfed and managing to stay in existence, the chloroplast, okay? And then it's also believed, this is a little bit more theoretical, but there's pretty strong evidence that the nucleus itself was a free living organism. So evidence for this that I'd like you to know, all of these organelles have double membranes. So the idea is it actually has the membrane of the original organism, and then when it was engulfed, that engulfing process put it in a second vesicle. So there's two membranes for the mitochondria, there's two membranes for the chloroplast, and the nucleus itself has two membranes. So that's all evidence for endosymbiotic theory. Each of these organisms has its own DNA inside. So it's got its own DNA, and that DNA is actually circular. So it's got DNA. Circular DNA is characteristic of bacteria. Okay, so in our cells, in animal cells and human cells, we have organelles that contain DNA that looks like bacteria DNA, and it's found in the mitochondria and in the chloroplast if you're a plant. So that's also evidence that it was a free living bacteria previously, or at least archaea. They divide independently of the host cell, so they reproduce on their own. The mitochondria will split and make daughter mitochondria. Then they move independently in the cell. They're semi-autonomous. And the membrane resembles prokaryotes. So the inner membrane, this inside membrane of like the chloroplast or the mitochondria, looks like it has the same kind of chemical structure as free living bacteria. So this is all evidence I'd like you to know that supports endosymbiotic theory. All right, that's the end of this video.